So one thing I'd like to point out about these splicing enhancer and silencer sequences is that mutations in those sequences can lead to human genetic disease. So about 20% of human genetic disease is caused by mistakes in splicing. And these mistakes can occur if a mutation occurs at one of the consensus sequences. We've already seen that for the beta thalassemia genes, or when a mutation occurs in one of the silencers or enhancer sequences. So a common mistake that is made when people are hunting for a particular, or scientists are hunting for the mutations that cause a particular disease, if a mutation is found in a coding exon, and that mutation changes the amino acid that's coded by that exon, it's often assumed that that mutation is having its deleterious effects by changing an amino acid in the protein. But in fact, many times the mutation could actually change the splicing pattern. So, for example, leave out a whole exon or change a splice site. And that itself would be much more deleterious. So it's important to think about that if you're trying to hunt for mutations that cause a particular disorder in either humans or some other model organism. And it's also important to note that we can't ignore mutations in introns. Not only are mutations at the consensus sequences going to be deleterious for splicing, but mutations in these exonic splice or intronic splicing silencers and enhancers can also change patterns. So in the future, we're going to have to be sequencing introns as well as exons when we're looking for mutations in human disease. So here are some examples of those kinds of mutations. The top here we have the SMN gene. Now all of us have two copies of this gene. There's SMN1 and SMN2. If you have a mutation in SMN1 such that it does not splice, or it is disrupted, this is a problem because the SMN2 gene is identical to the SMN1 gene except that it has a T mutation in an exonic splicing enhancer such that in the SMN1 gene all of the exon 7 is included, but in SMN2 only 20% of exon 7 is included. So if you're lacking the SMN1 gene, then you do not make enough of the SMN protein. And children who do not make enough of this protein have problems with development of their motor neurons and have spinal muscular atrophy. Here's another example. Dystrophin, there is a mutation which causes a stop codon, and so one might assume that the mutation, the problem is that it makes a truncated protein. But in fact, this stop mutation, this nonsense mutation, is in an exonic splicing silencer, or it becomes an exonic splicing silencer such that that exon is left out. And it still makes the protein, but it's lacking that exon. And then this is a third example. Patients that have frontotemporal dementia often have a mutation in the tau gene. And that mutation causes an exonic splicing enhancer to be stronger, such that they're making more of the version of the protein that has exon 10 included and less that has exon 10 excluded. And this 50-50 ratio is very important to have normal amounts of the tau protein. And so in this case, the patients are overexpressing this truncated form of the protein, and that leads to frontotemporal dementia. So finally, I'd like to leave you with my take-home messages. The first is that eukaryotic genes contain introns. Not every eukaryotic gene contains an intron, but certainly in humans, the vast majority contain introns. These introns facilitate the evolution of new genes, and by allowing for alternative splicing, they allow us to greatly increase our proteome complexity. And higher eukaryotes utilize exon definition to define splice sites, because the exons are like islands in oceans of intron. And then finally, many human hereditary disorders are caused by mutations that affect exon inclusion or splice site choice.